Okay, welcome everybody to this first GO2NE deoxygenation webinar. I'm Andreas Oschlies. I'm together with Marie-Laure Grégoire, one of the co-chairs of the Global Ocean Oxygen Network, GO2NE. And so it's an IOC UNESCO working group with the aim to better understand and make people aware of deoxygenation as a global ocean problem, and in an attempt to make life a little more pleasant and informative during these difficult times, we have had the idea to start this webinar in about, well, we will see how it evolves. We think of a monthly basis to tell each other our findings about deoxygenation, inform each other, give us a platform to communicate, to motivate early career scientists to stay in this field and move the field forward, and well, to be up to speed what is going on, and also to make provide a better platform, better knowledge base to inform society, and also to inform scientists who now target the UN Ocean Decade, I think, where we can, together with stakeholders, have a big input over the next decade. And of course, stakeholders are invited to the seminar as well, so it's open. And now I give, so this was the overall introduction about GO2NE and why we start the seminar. And Mike Roman is today's convener for the first ever GO2NE deoxygenation webinar. Thanks, Mike, and thanks to the speakers who are the joint guinea pigs today, and thanks to the audience for joining.
Well, welcome. I'm Mike Roman, and I'm presently at the University of Maryland Center for Environmental Science in the United States, and I'm a member of the GO2NE Scientific Steering Committee, and it's my honor to moderate today's seminars, which will be recorded. Our first seminar is by Dr. Karen Wishner. Karen is currently a professor of oceanography at the University of Rhode Island in the United States. Karen received her PhD from the University of California, Scripps Institute of Oceanography. Karen is a biological oceanographer with a focus on zooplankton ecology. Since the 1980s, Karen has studied zooplankton and oxygen minimum zones, including seamounts, the Arabian Sea, and the Eastern Tropical North Pacific. Karen is currently working on the Eastern Tropical North Pacific, which will be a subject of today's talk: deoxygenation in zooplankton, distributional strategies, and oxygen zone variability. And the talk will be about 20 minutes, and then we'll have 10 minutes of questions before we hear from Alexandra Steckbauer. Thank you very much. Go ahead, Karen.
Okay, I haven't received the note to share the screen yet. So I need to be, the presenter needs to be changed here. Okay, show my screen. And is that working?
Yes.
Yes, okay. People can see this.
Could you make it full screen?
Full screen. I think, let's see. Enter full screen. Okay, is that better?
Yeah. Perfect. Thank you.
What? This? What is going on right now? Okay.
You have little arrows down there to go back and forth.
I think I somehow got out of here. Okay, what are you, whoops.
We see you and we see your screen.
Okay,
we see your presentation.
I don't see my presentation right now. Let me.
No, now we don't see your presentation anymore.
Okay, let me get back to what this. No, but that. Let me get back to my presentation here. Okay. What? Just pointed at this thing? Okay, are you seeing the screen yet?
No, not yet. Now.
No, okay, well, let me start from the beginning. Okay, sorry. Is this full screen to you?
No, but it's okay. We can see the main features. Yeah, it's okay.
Okay, so thank you to the organizers for inviting me to this special webinar, and I'll be talking about distributional strategies and O&Z variability. I'd like to initially acknowledge the other PIs and co-PIs on this project, and these various projects over the years: Kendra Daly, Curtis Deutsch, Chris Roman, and Brad Seibel, and my technician Dawn Otrin, who sorted most of the copepods you'll hear about, and also our funding sources, specifically NSF for the most part, and our local institutions. So decreasing oxygen, as everybody on this webinar knows, is one of the major consequences of global climate change. These are well-known figures now in the literature, showing the OMZs of the world. We're working here in the Eastern Tropical North Pacific, and also the location of oxygen decreases during the past half century. These models, for obvious reasons, focus on the big picture. And today's talk, I'm going to highlight the small-scale hydrographic variability of oxygen minimum zones and show species-specific distributional responses to oxygen profile shape, oxycline depth, and oxygen concentration. And the focus is on calanoid copepods in the Eastern Tropical North Pacific. These are as examples of zooplankton adaptations to OMZs. So first, a little bit of the terminology for people who might be a little new to some of this. So these are the what I consider the OMZ ecological zones. These are unique ecological habitats with suites of zooplankton, specific oxygen levels, predators and food. In particular, this purple line here is the oxygen profile, and this is the OMZ core, the region of the lowest oxygen in the mesopelagic, and above that is the upper oxycline, where oxygen is increasing. Below that is the lower oxycline, and the actual depths of these zones and their specific oxygen levels vary with location. So this is an example of what fresh samples look like from these zones, and how you can identify zones just from looking at your fresh zooplankton samples. These are a set of photographs of samples, and arrayed in pairs, day and night, from the surface here down to 1,200 meters. I'd like to point out the OMZ core in particular, with very sparse during the night, a few more animals during the day when some of these fish migrate down into there. This is the lower oxycline, a unique community just at the base of the OMZ core. So our earlier work showed that looking at biomass as a whole for zooplankton showed that there were narrow biomass peaks, especially within the upper and the lower oxyclines. This is an extremely narrow peak at 2 micromolar oxygen, and that these boundary layers were typical of oxygen minimum zone regions. But today I'm going to focus on copepod species and look at species-specific vertical distributional strategies. So I'll show an example of each one of these: a mesopelagic oxycline copepod, a diurnal vertical migrator, a diapausing copepods, diapause as a hibernation part of the life cycle, and epipelagic copepods in the aerobic habitat. And just a reminder to people who aren't familiar with copepods, these are actually pretty small. They're really only a few millimeters, maybe from 3 or 4 millimeters long to maybe 10 millimeters or so, depending on the species. So first, a little bit about our sampling at sea. We use a mock nest. This has 8 or 9 nets and takes stratified or sequential tows. And we did day and night vertically stratified tows at a series of depth intervals, so we can actually array some of these tows vertically to get some reasonable definition for the vertical profiles. And we've also done some horizontal tows, that is towed at the same depth sequentially. And just to point out, those are the nets. This is the CTD that's now on the mock nest, and it's all computer controlled from inside the ship, so we can do adaptive sampling, track where we are. This is the when the samples come up, the cod ends are poured through a sieve. So these are the photographs you'll see of the fresh material in the sieve, and then of course they're preserved. So this is what we catch. This is again a typical profile from the lower oxycline zone. These little pink things are the copepods, and a variety of other taxa here. Fish. We identify the copepods to species by microscopy. Some 22,000 copepods, identified copepods, are part of the data set I'll be talking about. Over 300 copepod species. We're focused on adult calanoid copepods. And of course, I picked just a subset to illustrate some different vertical distribution patterns of this strategy. So we've had 4 different cruises, sampling at 4 different sites. Not every site was sampled on every cruise, sure. And you can see the locations are within this tongue of low oxygen water in the eastern tropical North Pacific. So the sites are CRD, which is the Costa Rica Dome, TB, which Juan Quebec Bowl, and then 2 other sites a little farther north. So first, a little bit about the hydrographic variability. These are vertical profiles, 3,000 meters. Each of these stations and years is color-coded. So for oxygen, we have 2 of these graphs, one showing the full oxygen range to the sea surface. The other one is truncated at 60 micromolars to emphasize this oxycline ONZ area. And the temperature, so you can see a lot of variability in oxygen among the different stations. The temperature, once you're below the thermocline, things are pretty similar for the rest of the water column, all of the stations. So we've taken subsets of these samples, the things in the black boxes, and then plotted oxygen versus temperature for the oxycline zone and ONZ zone. And just to point out, temperature essentially is equivalent to depth. So an animal that is staying at the same temperature or depth might encounter different oxygen values at different locations or times. An animal that tracks oxygen, stays at the same oxygen, might encounter different temperatures. To southerly stations, CRD and TB, there we had a very thick OMZ and a very narrow aerobic zone, very low oxygen in the OMZ, in the rough upper thermocline. In the 2 more northern stations, we had a much thinner OMZ. The OMZ is just very small over here, and a much broader aerobic zone, and gradual oxyclines, and the oxygen did not get as low. So first, the first copepod example I'm going to show is an example of a mesopelagic lower oxycline copepod. This is Lucifutia holzmanni. And to look at these graphs, up here are the different station abbreviations and the year. These graphs show the abundance for adults, the numbers per 1,000 cubic meters. In each graph, there's a blue line. That is the oxygen profile for that station at that time when those samples were collected. The gray box shows the OMZ core, the part of lowest oxygen in each of those particular stations and years. The red dotted lines are the daytime abundances, and the black points are the nighttime abundances. So for this particular copepod, it spends its entire life cycle at the lower oxycline. You can see that it's tracking oxygen, but of course that means its depth and temperature vary with location and change in these different areas. This one doesn't migrate. We know it's actively feeding and developing. It has a number of unique physiological adaptations, which people like Brad Seibel have investigated in this program, and it's living at very low oxygen, 1.7 to 5.9 micromole. And it's an indicator species of the lower oxycline. Just to show how all of the life stages are really intricately related to this lower oxycline habitat. This is actually from the Arabian Sea. This is a sibling species, a related species. You can see here the adults are living at slightly deeper depths and slightly higher oxygen. The younger stages are slightly shallower and at lower oxygen. But this whole life cycle is really coordinated with this lower oxycline habitat. So the next copepod I'm going to show is Metridia brevipara. This is a mesopelagic copepod with a split distribution and life stage separation by depth. So for this copepod, the adults and the older stages are mostly in the upper oxygen, as you can see. For these plots, we also looked at life stages for some of the locations, and so these bottom square plots are abundance of different life stages, color-coded by life stage. The top plots, just to remind you, are the adults only. So then the adults are in the upper oxycline, but the youngest stages are in the lower oxycline, and that suggests that they're developing at depth. And then for this station, the TB station, with its very extensive low oxygen region, the OMZ4, here you have the adults are very deep. Here they're at the lower oxycline, so they would be deep and cold. And but right up here at the surface are very abundant young stages, as you can see in these little bar graph plots. So clearly there seems to be some ontogenetic separation, migration to the surface. Our next example is a diovertical migrator, Floramama abdominalis. This genus Floramama, they're known for some strong diurnal migration. Now the expectation in much of the literature is that when you have a very thick OMZ, the diovertical migration is going to be truncated and their daytime migration to depth would be inhibited. Well, what we found was somewhat different. We found that the day depth stayed the same, about 300 meters, regardless of whether you had this very extensive OMZ or a much more gradual development of the OMZ. And this is likely determined by the normal predator avoidance and light penetration. It was the night depth that changed, which was somewhat surprising. So the night depth was shallow, only about 20 meters when you had this very abrupt thermocline and oxycline, and but it was deeper, about 100 meters in the stations which had the broader aerobic zone. And so basically more oxygen is available deeper. They migrate up at night only as far as necessary to obtain sufficient oxygen. Well, they were totally absent at the TB station in both years. We looked extensively through the samples for them. So perhaps when the oxygen is extremely low for an extremely broad vertical extent, that exceeds their tolerance of being able to inhabit that. And next is a deep diapausing copepod, Eucalinus inermis. So here, the adults occur either at the upper or the lower oxycline, quite a difference in depth range and temperature. And that where they occur varies with the profile shape. Now they're diapausing at these oxyclines in some of the stations, and we're diapausing at an oxygen concentration, very low oxygen concentration, 5.7 micromolar. And we know that they're diapausing because for some of these stations, we look at life stages and at the whether they had stored oil to get through their diapause stage, and high percentage, from 60 to 90 some percent of the older life stages in these diapausing layers had stored oil. So this is what those samples look like. So this is from the oxygen minimum zone, again, right up here. Just a little bit lower is this eucalinous layer, monospecific, when you catch it intact, with monospecific layer of eucalinous. And this is under the microscope, not the water column. You can see all the species of the animals. And then just below is this sort of lower oxycline community, again, institutional Holzman ion. So it's very distinctive in the water. So moving up to the upper water column, an example of an epipelagic copepod. So then a reminder, now this is 0 to 200 meters, so you can see the big difference in the amount of aerobic habitat. And so this is subucalinous subtenuous, an epipelagic copepod. And this copepod is abundant just above the thermocline, but it has an expanded depth range here where the aerobic zone is larger and the oxycline is more gradual. And as we interpret this as being an example of the aerobic habitat compression hypothesis, that the range is compressed when there's a narrow. So again, to summarize some of these vertical distribution strategies, the mesopelagic species are tracking oxygen. They change the vertical distribution associated with the OMZ intensity and depth of the oxycline inflection points. The diel vertical migrators adjust their migration depth to some point, especially the night upper depth. The diapausing copepods may shift the diapause depth at the base of the OMZ, and epipelagic species may expand or contract their depth range within the mixed layer associated with the aerobic zone. And because temperature decreases with depth, these changes in depth distributions have metabolic implications. So I want to go quickly through an example of one of our horizontal tows. So this, using a wirefly or deep profiler by Chris Roman, he was able to, it oscillates at depth, and he was able to do a 45-kilometer transect. And then we turn the ship around, and well, you can see that the colors are the oxygen, so you can see these sub-mesoscale features in that transect. We turned the ship around and went back through that profile with the horizontal Mach Ness tow. And this is what the hydrographic data looked like from that tow. So these were the different nets that were opened and closed, and this is oxygen, and you can see it's very small scale but consistent gradients. It's only between 0 and 10, or about 2 and 10 micromolar that the oxygen is varying. This is what the samples look like. So when you have the lowest oxygen, it's a pretty sparse sample. When oxygen is higher, you have more diversity and more abundance. And we're talking about an oxygen gradient of only less than 5 micromolar is our low oxygen definition for these samples. Greater than 8 micromolar is our high oxygen definition. So we're talking only about a 3 micromolar difference there, and yet you see quite a difference in the communities. So this is the quantitative information for a few particular taxa showing associations with the higher the low oxygen there in terms of their abundances in that transect. And then we actually did 3 of those horizontal transects and looked at these taxa, and we find significant differences in high versus low oxygen samples for most of these groups most of the time. So the conclusion, very small oxygen differences are matter to these zooplankton taxa. So why are these species-specific distributional patterns important? These changes in the depths of the peak abundance and diel vertical migration and the diapause depth affect mesopelagic food webs, all of those intricate connections from micro those affect many parts of the biological pump and carbon export, where you have your peak abundance of zooplankton, and affects a lot of those processes, and affects biodiversity and biogeography. And I'd like to just point you to some recent papers by Brad Seibel and Curtis Deutsch. It's a major component of this project, the metabolic modeling, which I don't have time to talk about, but they go into detail about the precise habitat requirements of some of these animals, and the metabolic constraints, how both oxygen and temperature limit the suitable habitat. Those suitable habitats are diminishing in the modern ocean, and there's really a limited scope for physiological adaptation to future change. So basically, we can use our understanding of variability today to inform future predictions. And this is just a little schematic of the processes that I've been talking about now in the context of the present to a potential future with OMZ expansion. And basically, I want to leave you with the points that the knowledge of the local OMZ structure and species-specific distributions are essential. We really need to look at this small-scale variability, and that I've just presented a few particular examples of patterns, but with from a few taxa, but unexpected responses and adaptations are certainly likely to be discovered dealing with more of these different organisms. So thank you very much, everybody, and I'm happy to answer questions.
Thank you very much, Karen. And we have time for a couple of questions. Here's one. Wait a second, it just went away. Hold on. Are the organisms most sensitive to oxygen concentrations, oxygen partial pressure, or oxygen partial pressure? Is there a significant difference among the temperature and oxygen conditions sampled, or temperature and pressure?
We certainly sampled across a wide range of oxygen and temperature conditions, and once again, Brad Seibel and Curtis Deutsch are really the modelers for that. But yeah, the animals, these taxa have very specific temperature and oxygen habitats that they seem to need. Some are more broader, arrayed in a more broader vertical range or habitat range. But that's one of the future issues, that these environments, the specific combinations of environments, temperature and oxygen, are perhaps being reduced in the extent of the geographic ocean as OMZs expand.
Great. That was a great talk, Karen. I enjoyed it. I have a question. So I know you sampled the adults. What about the juvenile forms, the nauplii? Are they, are they hatched at the surface? Are they down in the OMZ? Do you have any inclination of where the juvenile stages are?
We for some of the animals, as I showed, we certainly looked at copepodite-like stages. The netmesh doesn't really get the nauplii. So I don't know. Some other people have certainly sampled some of the Calamus enormous nauplii and several other types. But yeah, that's a very open question. My suspicion for the Lucifutia holzmanni is that their entire life cycle, including the nauplii, is down there in the lower oxycline. But clearly, a number of these other taxa have moved up and down. Parts of some of their life stages are shallow, some deep. So it's still a, we didn't have a seasonal pattern of sampling at all. So we can't really address all of that. But yeah, basically we don't catch the nauplii.
Gotcha. Here's another question. While this is an environment with very low oxygen conditions, even anoxic in some cases, there is also a corrosive environment with very low pH, high PCO2, and low calcium carbonate saturation. How do you think oxygen interact with such a critical parameter such as pH on copepod physiology? And There's a second part, but I'll let you answer the first one first.
I think the next talk is going to combine the pH, ocean acidification, and oxygen set of things. We do have, there's some interesting work by Amy Moss, for example, in one of our earlier cruises, who showed that a number of the species of pteropods actually do have diel vertical migration into some of this very low oxygen water. So even though they're thought to be extremely sensitive to acidification, they at least are there for parts of the diel cycle. We also, Kate Davis at Yale is now looking at forams, pelagic forams from our samples, and she finds the unique foraminiferan species, a particular species that is characteristic of the OMZ water, and she's hoping to develop that as a proxy for OMZs in the sedimentary record. She's a combination geologist, biologist. So some animals do have different responses, and some are surprisingly able to deal with that habitat.
Okay, one last question, and then we have to switch over to hear Alexandra's talk. You mentioned the changes from 8 to 5 matter for zooplankton. What is the level of accuracy that you would recommend for oxygen data?
Well, I guess that's a hard one. Certainly some animals undoubtedly can live at a wide range of oxygen, but I think we're seeing that. I would say you have to measure to the micromolar, and we didn't go to areas that got anoxic. Certainly some of the areas of these oxygen minimum zones we go to non-measurable amount of oxygen. So, and of course the microbes are dealing with these very precise oxygen values as well. I would say for the zooplankton, at least the types that we measured, certainly down to the micromolar level, we need to understand that variability. It was surprising to us to see as much variability in abundance and diversity at this very small oxygen gradients.
Okay, well, great. Thank you very much, Karen. And again, this will be recorded. And if you see on the last slide that Karen had, it lists her email. So if you have any further questions, I'm sure she would be glad to entertain them. So just send her an email. Okay, well, thank you very much. Let's
Alexandra, I will make you now a presenter, and then you can show your slides.
Great.
We can see
Welcome.
your screen. Yes.
Can you see the presentation?
So far, we only see your screen, so you have to go on the PowerPoint. If you have to,
No.
and now we see the presenter mode. You have to go to the 2 displays there. Just go to your PowerPoint.
Yes. Wait, I can just switch it over. Then it should work.
Yes, and now go to the 2 displays because because you have the presenter mode. Yes.
Yeah.
No. Yeah. Make it full screen. And then you have the black.
Here.
And yes, exactly. Exactly.
Oh, okay. Sorry.
Boy, that's great. Well, welcome, Alexandra Steckbauer. Alexandra is currently a postdoctoral fellow at King Abdullah University of Science and Technology in Saudi Arabia. Alexandra did her bachelor's and master's degree in Austria at the University of Salzburg in Vienna. Alexandra received her PhD at the Universidad Internacional Menéndez Pelayo in Mallorca, Spain. And she studied a variety of anthropogenic stressors in coastal environments, including the ecosystem impacts of hypoxia, as well as the synergistic effects of both hypoxia and ocean acidification. She's currently working on how multi-stressors affect Red Sea coastal ecosystems in the lab of Professor Carlos Duarte. So it's a great pleasure to introduce Alexandra, who's going to talk on defining oxygen and CO2 syndromes in the ocean and how deoxygenation and acidification impact marine biota.
Yeah, thank you for the nice introduction, and thanks for the invitation. It's an honor to be here today. Do you also have my picture big on the screen? It looks good.
So we see your
on top of my
presentation.
so I cannot see my own slides. No.
We see you and your presentation. Okay, I don't know what.
So thanks again for inviting me today. And today I will be talking about two of our recent publications with my colleagues, Dr. Shannon Klein and Professor Carlos Duarte, on the terminology on oxygen and CO2 syndromes in the ocean and how deoxygenation and acidification impact marine biota. First, we heard now from the OMCs in the ocean, but now I want to go more to coastal ecosystems. So the first hypoxic zones have been observed in 1985 in Louisiana coastal waters. And hypoxia is usually defined as a threshold of dissolved oxygen in the water of 2 milliliters per liter after Diaz and Rosenberg, which equals to 2.85 milligrams per liter. But depending on the current or ambient conditions, different authors use different concentrations, for example, as well, 2 milligrams per liter, which is much lower. In coastal ecosystems, hypoxia is usually favored due to high temperatures, which favors high respiration, semi-enclosed water bodies and estuaries. And Diaz and Rosenberg in 2008 presented a first map with the location of these hypoxic zones, which was updated in 2017 by Altieri and colleagues, which we see here. As you can see, most of the studies have been or most of the locations are in the Northern Hemisphere. And with this new map, there were more in the Southern Hemisphere. And what's new is, which was missing before, are the yellow dots, which represent hypoxic observings related to coral reef mortalities in the ocean. Also important from a meta-analysis reveals that the hypoxic coastal sites are increasing exponentially by 5.5% per year. And this will be ongoing and is predicted to continue in the future due to ocean warming and eutrophication. These authors also looked at the species and the oxygen limits, and they found that the 90th percentile of the super threshold is 5 milligrams per liter, so much higher than the thresholds previously proposed, and the median lethal concentration is 4.6 milligrams per liter. I would like to take the opportunity and talk to a related topic, ocean acidification. As we know, CO2 and oxygen are linked in the biosphere and also due to respiration and production in the water. And with the emissions since the industrial revolutions, CO2 in the atmosphere increased, and it is predicted, depending on the different RCP scenarios, from 2.6 to 8.5 to increase further in the future and might even reach 700 by end of the centuries. The average ocean pH already declined by around 1 unit, and it's predicted to decline further by 0.14 to 0.41 units by end of the century, depending on the different scenarios. Due to the increased CO2, it's lowering pH in the water and also impacting the organisms, for example, respiration and calcification rates. So for our first publication, I want to present, we dived into the literature and we looked around terms that have been used previously to describe drivers and events of deoxygenation or reduced oxygen. So obviously we have hypoxia, but it also has been called dead zones by Diaz and Rosenberg, because Usually they observed, for example, fish kills or mortality events, also marine benthic hypoxia. Then we have ocean and coastal deoxygenation. It was also called oxygen depletion, eutrophication-driven deoxygenation. And similar for the acidification, it was referred to as elevated CO2, ocean coastal. But authors also get really creative when they try to describe their local conditions as close as possible. For example, dial coral reef acidification or coastal ocean. And this includes us as well, because in different studies we used sometimes different terms. We also looked at classification of this terminology and these ecosystem processes, and if there are definitions available. And the only one with that definition is ocean acidification, which was made available by Gattuso and Hansen in 2011, while the others have just a general description made available. For the ecosystem states, they are usually referred to as elevated CO2, and for oxygen, reduced oxygen, hypoxia, anoxia, for example. And on an organismal scale, there are different terms that are used, for example, hypercapnia and acidosis and hypoxemia. These terms usually are referring to blood or body fluids and are also used in different fields like biomedical research. We also looked at, did an attribution analysis to look what are the drivers of these different, if we look at coastal and ocean separately. And for the ocean, the main drivers are reduced solubility of the oxygen due to warming-driven oceanic outgassing, but also anthropogenic aerosols. When we go to the coastal zones, the main drivers are primary production, respiration, and oxygen consumption, so just to name a few of them. Similar for the acidification, the main driver are air gas exchanges and biochemical processes from the atmosphere. Whereas for the coastal areas, we have circulation of waters, buffering capacity, also fresh water, and obviously also dissolution and calcification processes. So to get all these different terms that have been used, because they also have been used interchangeable and caused some confusion as well, we present a workflow to separate between rising oxygen and decreasing rising CO2 and decreasing oxygen values on a global versus local scale. So the nature of the driver would be global climate change versus coastal processes, including watersheds, metabolic processes, but also climate change. The onset and duration can be persistent, so it takes decades to centuries, where in coastal ecosystems it can take days to years, but also then expand to decades and centuries. As I said, usually climate change is globally, and whereas in coastal are more impacted on a local to regional scale. Very important is also the severity of the stressor, where we have for the global scale the IPCC RPC scenarios. As I already mentioned, the decreased range of pH is predicted 0.14 to 0.43 for pH, and a decrease of 1 to 4% for oxygen, depending on the scenarios. Whereas in coastal ecosystem, we can have moderate to very extreme fluctuations or decreases in both pH, and oxygen can go down to anoxic in very short time periods. So to separate them out, we are proposing to use ocean and acidification and deoxygenation, and separate that out from coastal acidification and deoxygenation. As I mentioned, there was already a definition made available from Hansen and Gattuso. For the definition, we added the word global to now read the global reduction in pH of the ocean over an extended period, typical decades or longer, and primarily caused by the physical uptake of CO2 from the atmosphere. And we provide a similar definition for the ocean deoxygenation. As for the coastal zones, we also now provide or propose definitions as the decrease in oxygen or pH of coastal systems and or the adjacent ocean systems over periods ranging from days to decades. caused by global processes and ranging from local to regional scale, which includes watershed metabolic processes and, for example, eutrophication. We acknowledge, however, that there are scenarios where, for example, nutrient inputs can lead to a reduction in oxygen in the coastal waters, which can then impact the adjacent ocean systems. And reversed, there are processes like upwelling that come from the deep waters due to warming, and they can also impact then the local and regional coastal waters. So to summarize this first part of the presentation, both open ocean and coastal processes have different attributes and drivers. And we think it's really important to be consistent with the terminology, not just within the research community, but also to make it easier for stakeholders, politicians, and the general in public. And we hope that these definitions and clarifications provided will improve discussions, but also the communication between or within the research community. For the second part of my presentation, we conducted a meta-analysis. So to bring together this oxygen and CO2, as I said, they are linked in the atmosphere. The respiration index was proposed by Brewer and Pelzer. This is a simple linear constraint which brings together PO2 and PCO2. They also propose thresholds of 1 would be the aerobic stress zone, 0.7 the practical limit of aerobic respiration, and 0.4 was the practical dead zone where nothing was, aerobic life was not possible. But this respiration index has also been heavily criticized, and Brewer and Seibel and Childress said that not only is the RI invalid, but its use also leads to incorrect and misleading predictions of the threat of changing oxygen and carbon dioxide in marine life. But in fact, it has never been tested. To test that, we calculated some theoretical combinations of the PO2 and PCO2 for ambient waters and then deoxygenation and acidification alone. But if we combine the new stressors, it can get really severe and we can get really low values. In recent years, there was a review published by Gobel and Baumann in 2016 on the impacts of hypoxia and elevated CO2 on marine biota, and they looked at 8 full factorial studies. And they found that the majority of the results or responses were either additive or synergistic. So the aim of our study was to assess the potential interactive effects of the reduced oxygen and elevated CO2 on biological responses of marine taxa, but also to test this previously challenged respiration index, if it can be used or if it can be a potential predictor in the future. So we used a meta-analytical approach and we conducted a literature search, and we retained publications that used and assessed the biological responses in a full factorial studies. This was really important because we needed both individually and in combination to assess the interactive effects. They also needed to provide sufficient data for the description of the water chemistry, so we could calculate PO2 and PCO2, and they also needed to report mean values, sample size, and a measure of variance. So overall, our data set, we found 363 experimental comparisons from 16 individual publications. The response variables included respiration, survival, growth, and metamorphosis, and others. But we also found a huge lack of data for tropical regions, as most of the studies were from temperate and subtropical regions. And there was also a gap of knowledge for some taxa as echinoderms and crustaceans. If we look at the variation in experimental treatments, to simplify, we used the terms acidification and deoxygenation as treatment terms here. For the control, we used whatever was defined as ambient or controlled by the authors, and we did not limit this data set to or cut at any specific value. For oxygen, our threshold was 4.6 milligram, which is the median lethal concentration, as I mentioned before. And our lowest value was 4.06 milligrams per liter and did not overlap with there was no overlap between the deoxygenation and combination treatment versus the control and acidification. And what was interesting was for the CO2, we had a high variability of what was used by the different authors, and our highest value was around 3,500 microatmosphere. To standardize our biological responses, we calculated the effect size, the variance of the effect size, and also determined the confidence interval of significance after Hedges and Gurevich. And we also determined the potential of interactions, where we calculated the main effect of the individual stressors and the effect of the dual stressors to see if the impacts were additive, synergistic, or antagonistic. Synergistic interaction was when the effect of both stressors is greater than the expected response from the sum of the individual ones, and antagonistic if it was smaller. So if we have a look at the results, we plotted the effect size versus the PO2-PCO2 ratio, the respiration index, and we can see that there was a huge overlap between deoxygenation and the acidification results. But if we combine both stressors, the impact is much more severe. If we look at on a taxonomic level, here we can see that most of the results or impacts were negative. The full circles are where the confidence interval does not overlap 0, which means the results were significant. We also calculated the addictive effects and compared to the observed effect of the combination of the two stressors, and found that for most comparisons they matched. If you look at the box on the right, you can also see the overall results, which were all significant from each other. And overall, we found that 91.7% of the responses were additive, 6% synergistic, and 2.3% antagonistic. And we also calculated 0.1 bins to get an overall trend versus the effect size, and we also calculated the reduction. And if we compare this to the limits that I mentioned before, we can see that the criticism is valid because the thresholds are completely off, and we even had at 0.2 even specimens that were alive. So to summarize, we had predominantly additive but variable impacts. The PO2-PCO2 ratio provides a useful continuum to assess the impacts of these dual stressors, but the criticisms are confirmed as there was an overestimation. And we also show, which is really important, that experiments targeting only one of these stressors might not show the full picture. And what was really interesting as well, that there is a huge gap in knowledge, as I mentioned, for some taxonomic groups as echinoderms, crustaceans, and anthozoans, for example. None of the studies was done on corals, on calcifying fires, tropical regions, and also responses that were measured. Okay, thank you for listening. And if you have any questions, ask ahead, or you can even also send us an email if you want.
Boy, that was a great talk, Alexandra. Thank you very much.
I just switched it off.
While people type in their questions, I have one. So, you know, the multi-stressors of low oxygen and high PCO2, there's a third variable that could affect the differential response, and that's temperature. So, of course, when temperature goes up, the animals have a higher respiratory demand. So even at the same oxygen level, they will be stressed. I don't really know how temperature would affect acidification effects, but if it's different than mathematically than oxygen, it could potentially affect the relationship, yes?
Yes, that's true, but unfortunately, all of the studies only had the ambient temperature. None of them had a temperature as a treatment, a third stressor included.
Yeah, I wonder if you could use like a Q10 on the respiration, or might be some way to
Exactly.
them all, but yeah, that was great.
None of the studies included temperature as third stressors. But there are more and more studies coming out, so hopefully in the future we can fill some gaps.
That's great.
More questions?
Okay, here's one. Well, they're all saying great talks. They're saying, are there organisms most sensitive to O2 concentration? Oh, this is for, I'm sorry, that's for Karen's.
Sorry, Mike, in the chat box, I put the questions which are.
I think there's one.
Alexandra, great talk. Thank you. What would you suggest to use instead of RI as a metric? Understandable to various stakeholders, could simplify communication, similar to the terminology paper you first presented.
Instead of the RI, that's why we tested it, because that's to the best of our knowledge there, or so far the only thing to bring both together and actually get them on one matrix, because otherwise you can just use one or the other, but to bring this combination together in one figure, for example, it still holds, it's still a useful tool. But obviously in the paper we acknowledge that depending, as I mentioned, depending on the combination of PO2 and PCO2, you might get similar values. That's why with individual stresses, you also have this overlap in results.
Yeah, and of course, in coastal waters, many climate, depending on where you are, IPC scenarios have more rainfall. So with the rain coming in, you know, that Weijun Kai has shown, you know, that will, with the humic acids, that will, you know, affect the water pH as well. So that's an added stressor, you know, with salinity that you can do in your next paper.
Then eutrophication, yeah, that's why we put it on local to regional scale, because often you have so many different stressors or like local impacts that a reef further or a location further you might not have.
Right. We have just about 9 o'clock. That's all the questions, but I'm sure that, you know, with, again, looking at the recording, and you can send that to your friends if you have any questions further for Alexandra. I'm sure she'd be glad to answer them. You know, this was a fantastic start to the seminar. We had, I think, the peak 327 people attend. So this speaks to the interest that people have in global deoxygenation. So I'm going to turn it back over to our chair, Andreas, to say goodbye to you. But yeah, thank you so much. It was a great kickoff to the seminar. So I want to thank personally Alexandra and Karen for doing a great job. I really enjoyed the seminars.
Yeah, thank you for organizing, and thanks for the invitation. It was really good to be here.
Yes, thank you very much, Alexandra, Karen, and Mike, for this first series of the GO2NE webinars, and thanks to Kirsten and the IOC team for all the technical support. And thanks to all the attendees to make this happen. So on behalf of the GO2NE team, Marie-Laure, me as co-chairs, and the entire team, we well, thank you for this successful exercise, and we would like to announce already the second one. So after this first success, we can safely advertise the next one. Will take place on 9 December 1 hour later than today, and Laure Resplendy and Søren Thomsen will be the speakers. And if you want to be informed, well, register at the IOC or GO2NE website and share the word. We can still accommodate a few, and I hope that, well, anybody who is interested giving a presentation, please contact us. So this should be an exchange platform to make us aware of your most exciting novel research, and also give you the opportunity to connect to the growing society of people interested in deoxygenation, ocean oxygen, and educate us, since there's this lack of communication right now during the COVID crisis. And so we want to move this forward. The science is still important, and deoxygenation doesn't go away so quickly, so we have to move forward. Thanks very much for all your initiative, your effort, and your participation in this great event. Goodbye. See you soon.
Bye. Thank you.
Thank
Bye.
you, everybody. Bye.
Bye-bye.
Look out.